guys for being here. I'm excited to introduce Jon Puritz, our speaker for today. Just a little background. So he started his undergrad at Brown with John Whitman and David Rand, and kind of like discovered his interests in biology and science with research with lobsters and pesticides. From there, he then did his PhD at University of Hawaii with Rob Tunin. And actually was like the one person in his department not doing work in Hawaii systems. He did some work out in the Pacific Coast in Australia with bat stars in Southern California. And then he did several postdocs at Texas A&M and then also a postdoc at Northeastern, basically building up bioinformatics skills and kind of figuring out the oyster system that he now works on today. And then finally in 2017, got his position at University of Rhode Island, where he's working with the pool oyster system using lab experiments and genomic techniques. And I'm excited to hear more about your stuff today. So thank you.
Thanks, Maddie, for that introduction. Thanks everyone for having me. I hope to give you a nice 45-minute break from existential dread today. I do want to start just by acknowledging that the University of Rhode Island occupies the traditional grounds of the Narragansett Nation, and all the places that we do research, whether it's like field collections right now or laboratory work that I'm going to talk about, is all on those lands. And so I want to acknowledge that before we get started. I also want to acknowledge my lab and all the people in it. You'll see a lot of the work that we do takes huge amounts of efforts from lots of different people. And most of the time they're all doing it with smiles on their faces. And they've been a great group. And I will say we a lot, and it's the royal we of them doing a lot of the work that I'll be talking about today. Okay, so this is going to be a really easy audience to do this introduction. I think it's really easy to convince folks, that we're now living in this new geologic age, the Anthropocene, and that human impacts are really one of the predominant forces that are affecting the entire globe. And it doesn't matter what ecosystem we talk about, but we have this extensive, rapid change that's happening basically because of the explosion in human population and technology that's happened in this time. And it's really easy to trace that on a graph. And this has really led to amazing changes in biodiversity that we've had across different ecosystems. This is a fairly old paper now, but they did some really great modeling showing sort of, what extinction rates were in the distant past, in the recent past, and then modeling in the future. And as far as I've checked, we keep heading more and more towards that projected rate of extinction, where we're basically going 1,000 times higher than would be expected, sort of based on our background. And this is, coupled also with global declines in genetic diversity. And, I think I don't have to convince all of you why that's important, but, for everyone else, loss of biodiversity leads to degradation of ecosystem services. And I think that's especially true in the ocean. And it's not just me that thinks that, but this is something that's, of great concern, I think, to everyone. And just to make things broader, I think this is a stunning graphic that came out of a science paper, and it's been highly publicized, but that's a big number of loss, and that's birds since 1970, 2.9 billion. I mean, that's staggering. And more things about billions of things disappearing. I mean, the Alaska snow crab 2 years ago, 11 billion disappearing. I mean, I think it's just like this a crazy time to think about how we're affecting biodiversity and the changes that are happening kind of at these rates. And if we factor in what's happening with climate change, that picture just gets kind of scarier and scarier. And so to me, like a big motivation of the research in our lab and a motivation for me as a scientist is to understand, both the natural processes that generate and protect biodiversity and like how we as a species, how human beings are affecting those processes. So this is like sort of the giant motivation of what's going on the research in my lab. And for me, which will be very familiar to you, I very much equate genetic diversity as part of this biodiversity. So I'm really interested in processes that regulate genetic diversity, specifically population connectivity and selection. And I don't have to explain either of those terms to all of you, which is great. So really research in my lab falls under two broad categories. The first one is developing genomic tools to ask evolutionary questions. I never wanted to be a methods person, but I keep finding ways to develop more methods to my chagrin. But basically, this is taking advantage of next generation sequencing technology and bioinformatics to build these tools that we can use to answer exciting evolutionary questions. And that's sort of the second part of what's going on in our lab, is really understanding how both the environment and humans are impacting the evolution of marine populations. And we do this by looking at population connectivity, looking at natural selection, and trying to put that in some sort of seascape genomics framework to try to understand how that's all connected. And then I like to take these little 5 things and put them into a 5-pointed star, which gets to my favorite marine invertebrates, also the basis of our lab logo. So this really sort of pictorially encompasses what we do in the lab. So today I'm going to try to hit upon a couple of those things within our 2 broader categories to share with you. The first one is I'll talk about this new method of exome capture called EEC-seq. I hate to start with a methods talk. I promise you I'll keep it interesting, but everything else is based off of EEC-seq, so it's really critical that we start there. And then we'll move on to some more interesting stuff, looking at how a couple different coastal stressors are affecting the evolution of species, which is the eastern oyster in Narragansett Bay. So, most of you are very familiar with Crassostrea chagas, but Virginica serves a lot of the same really important ecosystem functions. It's a habitat creator. It's an amazing water filter. It's also a shoreline protector, building up reefs. And lately, it's become a very important economic influencer. Aquaculture has been a booming industry now, especially in New England. And so there's a lot of interest in oyster populations and what they're doing because it's one of the most carbon neutral animal proteins that you can get on the market. Oysters are a really exciting system too because you have this really nice pairing of a biphasic life cycle where oysters as adults don't move. So that's a really nice tractable system. And then we have this nice 25 to 30 day pelagic larval duration that's connecting our different populations. And I didn't really know about this, or I didn't think about it too much, but I may have been destined to study oysters from a young age. This is me at age 6, dressed up as my favorite character from this book called Ula Oyster. And yeah, I never thought I'd be doing this, but here I am studying oysters. And oysters also exciting, not just for this life cycle, but they basically occupy this entire coastline. So from, basically the very bottom reaches of Texas all the way up to Canada. So amazing environmental gradients and lots of different potential for adaptation. So a really cool system to work in. So I like for long talks to start with my take-home messages. So hopefully today I'm going to convince you that EEC-seq is an effective tool for understanding adaptation, that coastal stressors shape larval genomic diversity, and that understanding these stressors across multiple spawns is really important in experimental context. That coastal stressors also affect the distribution of adult adaptive loci, and that looking across life history stages is critical to understanding selection on marine species. And hopefully I'll get to convince you of all 4 of those points. And so, like I said, I'm going to start with the methods talk, and I apologize for that, but this is a method of exome capture that I developed originally as a postdoc in Katie Lauderhouse's lab. So just to review, the exome is all of the exon loci in a genome, so all of the protein coding regions of the genome. And I'm not going to go over the central dogma here with you, but I'm sure you're all very familiar with this. But sometimes what you might not realize is that the protein coding portion of the genome is actually a really small portion of the genome. So this is actually color-coded by scale for the human genome. It's about 2%, 1% of the genome size is actually protein coding. In oysters, it's a little bit bigger, so we can make it this size, just sort of using this as a graphical representation. And so I'm always sort of thinking about these questions from a population scale, are we looking for different loci and different changes in frequency? And sometimes you're doing this in very small portions of that, even that tiny portion of the genome. So just to kind of illustrate this, like imagine we have 2 oyster populations, because that's what I work on, or in some sort of magical environmental gradient, and that, we may have 2 different loci, like just say like 2 different SNPs. So I know they're very small and hard to see, but I do that on purpose because that's basically the finding exposition that we're going on all the time. And when we have to, try to find small differences in allele frequency between different populations, you want to be able to sequence a lot of individuals. And one of the ways that you can do this, if you're, dealing with realistic scientific budgets that we're all dealing with, is having some sort of method of genome reduction. This allows you to stuff more samples on that sequencing lane and hopefully get at some of these more accurate characterizations of allele frequencies in populations. A lot of you, I'm sure, are familiar with different ways of reducing the genome down to do this, but I just wanted to go over a little bit of a review and sort of compare and contrast. One that's been very popular for a long time is RAD sequencing. And RAD sequencing is really accessible, easy to use. There's some well-established pipelines out there to do the bioinformatics. The trouble with RAD sequencing is I'm just going to use these little bars to sort of represent hypothetical reads coming down on the genome, is that you're sampling at fixed locations, and those locations are quasi-random across the genome and fairly widely spaced. So if you're looking for genes under selection, you may not be actually capturing that variation, or may not be able to capture it with the enzymes that you're using. So even though this is cheap and really accessible, it may not be the absolute best thing if you're purely interested in selection. RNA sequencing is another really popular way for doing genome reduction. And this is a great way because you're focusing all of your sequencing effort on protein coding loci. So that's really great. The problem is that we have differential expression, and you end up wasting a lot of money on reads that go to these really highly expressed genes. So it can be somewhat expensive if you're just trying to scale the populations. Another issue that can pop up with RNA sequencing is that you may run into this phenomenon called allele-specific expression. So this way, you may just have one allele from the genomic copy being expressed. And so if you're tracing this back to try to infer allele frequencies in populations, that can be a little problematic if you're not seeing that in your expression data. So exome capture kind of offers a little bit of a happy medium where you can sort of target all of your reads in the genomic DNA and in the genomic portions that are protein coding. Now, this is me showing bias. This is not exactly how it works. Really, you have like about 50% efficiency with exome capture. So you do spread reads across the entire genome, but you really focus the vast majority of them on the protein coding sections. The problem with exome capture beforehand is that typically you needed to have a genome, you need to have a transcriptome, an idea of where these protein coding loci were in the genome, and then you had to synthesize probes. So you had to send off a lot of money to a company to get you these probes. So basically, just to iterate that, you had to put in a lot of time and money and send it to either microarray or MyBates to get this stuff. And then you would go through a standard procedure of doing in situ capture to do your exome capture. And so we kind of had the idea of like, well, maybe we can cut out the middleman and have our organism do this process for us. And so we've been calling this EEC-seq, or expressed exome capture. And what it boils down to is we use mRNA to probe and bind to the exon loci in the genome. Yeah, I've been calling this colloquially EEC-seq. So to sort of test this, we did a really simple heat shock exposure, just really to get some expression profiles that were predictable. So we cooked some oysters for an hour at 36 degrees Celsius compared to the ambient 12 degrees Celsius for an hour. And so I'm going to represent that with we have ambient and hot oysters. And I'm about to show you, I promise, the most complicated PowerPoint slide you've ever seen, but we're going to go through it really slowly. And hopefully you'll be able to understand how this procedure works. So with your study organism, with EEC-seq, what you want to do is be able to extract both RNA and DNA from different subsets or the same subset. So the first thing you do is you start with RNA extraction. Get your RNA, that I'm going to represent here. And then you go through a standard mRNA library prep we've been using the Kappa kits to do this. The one difference that we make is we have custom Illumina adapters that we've added a restriction site to the very end of it, or I should say the very beginning of it. After that, we go through this process called duplex-specific normalization, and that's really to help get rid of some of the really hyper copies that are in that transcriptome. And you can even save that before or after normalization if you want to sequence the transcriptome. And after the normalization, we then remove the adapters with the restriction enzyme, and then we add this molecule biotin to cDNA with an enzymatic reaction. And so that becomes our probes. Then you go and you extract DNA, starting again from the beginning. And here we have DNA. We're going to just go through a regular shotgun library preparation. And here I'm going to color code exon loci in blue and non-target loci in gray here. So you get this. So this is just standard. You have all that stuff in your lab already. And so you have your genomic library and the probes. And what we do is you hybridize them together in a tube. And during that hybridization, basically what happens is we have the probes hybridized to our target loci, and the non-target loci does not hybridize. And so this is where the exon loci are being hybridized. Then we go through the process of capture. And to do this, we take advantage of the fact that streptavidin and biotin have a really strong chemical affinity for each other. And you can buy magnetic beads that have streptavidin coated on them. So we use this to isolate and capture those probes with the exon loci hybridized to them. You go through a few different washes and elutions to get rid of the non-target ones, and then finally elute off your target loci. Go through a few cycles of PCR enrichment just to boost up your numbers, and then you do exon sequencing. So yeah, promised you, we made it through, right? So really, one of the advantages here is that you're targeting things that are being expressed. And so just to highlight this, I'm going to zoom into a part of the Eastern oyster genome. So here's a length-long chromosome, and this is going to be sequencing depth, and we're going to go both in the positive and negative polarity. So here I'm just going to mark genes across the genome. So this is a heat shock protein with the orange representing coding sequence. And here is a gene that I'm blanking on the name of what it is, but it wasn't something we were expecting to be expressed during our little heat shock exposure. So here in the negative direction is actually coverage from sequencing the transcriptome probes that we used. So here we can see we get highly expressed heat shock protein, is what we expected. And we have very little expression at this other gene here. And this is on top is the DNA coverage that we're getting from the captured DNA. So here we're getting really nice coverage at the same loci that are being expressed, and we're still getting capture even at some of these lowly expressed loci. So we are really enhancing our coverage at these protein coding parts of the genome. So I'm happy to answer more questions of that on the end, but hopefully I've convinced you that this could be an effective tool. And right now I have a PhD student in the lab, Jacob Green, who's working on really trying to make this a method for all organisms. We're going to have a de novo bioinformatic pipeline so that you really don't have to have any genomic resources to do this. And we've been doing some trials to sort of make it as efficient as possible, because as you saw, there's a few different lab steps, but we're trying to make it as easy as possible to do. The protocol is open and online. The publication proving the concept has been published for a while now, and the de novo bioinformatics pipeline will hopefully be out, I'm hoping, by like next spring. I think we're getting close to that. So this is a method that we'll use for the rest of the talk. So I hope that I've convinced you it's a good one. But now we can kind of move into the fun biology stuff, and this is going to focus on these impacts of multiple coastal stressors across different populations. So I'm going to come back to our hypothetical 2 oyster populations. I'm going to put this in a little bit more of an environmental context. So we'll put some city and industry and sewage treatment, agriculture, just sort of a representative picture of the environment and how humans have sort of changed the natural environment of, say, an estuary that we might experience. And we can think about all kinds of different stressors that might be here. Carbon dioxide, acidification, effluent coming in from the treatment plant, low dissolved oxygen, impacts of eutrophication, so excess nitrogen coming in, salinity stress, microplastics, temperature. I could just keep making funny little icons that put on here, but it's a complex and stressful world in these coastal estuaries. And one thing I want to spend a little bit of time on, because this is different on the East Coast than it is on the West Coast. I know probably, lots of folks, when they hear ocean acidification or coastal acidification, you might think about, downwelling and upwelling dynamics. I'm sure that's what you're thinking about here on the West Coast. If you're thinking like broad oceanographic, you might be thinking about, just atmospheric carbon dioxide coming from, us and then going into the ocean. But on the East Coast, where we have the eastern oysters are living, a lot of this acidification is driven actually by nutrient inputs. And basically, what we get is eutrophication coming in, and these nutrients provide blooms to phytoplankton and microbial communities, and then through community metabolism and respiration, basically the nitrogen increases, the oxygen decreases, and the CO2 increases. So we have this coupling between periods of low dissolved oxygen and high CO2, so acidification. And this varies seasonally and on a daily basis. So this first subpanel here is just the trace of oxygen and pH across months. And then here is an example of a week in the winter. So this is February, and you can see that it does go through diurnal cycling, but the variation is not that strong. In contrast, this is in the summer, and you can see very wild swings in both pH and dissolved oxygen. Almost in some places, you'll get 1 pH point of swing in a day. So this is a really dynamic system, and it's a very prominent driver of acidification on the East Coast. And so these, like I said, these are both mechanistically linked, and it's been shown by modeling that basically the more we increase atmospheric CO2, the likelihood of increasing this amplitude and strength of these fluctuations will increase. So it should get worse over time. So right now in the lab, we have a couple different projects that really focus in on this paired stressor, and where we add in another stressor just to make things complicated. We have one project that looked at sewage effluent, and one that looked at low salinity in combination with these stressors. And all of the projects really work to try to bridge between these life history stages, with the thought that some of these early life history stages might be sensitive to these stressors. And just to sort of hit home to the point, when we're dealing with benthic adults, our larval exchange is the only way the genetic exchange is happening between populations. So it's really critical to understand how these early life history stages might be happening. So, you might be in a situation where we have stressors coming in, and this is acting as just a full-on barrier to dispersal, where everything just gets, is dead, doesn't get across to that. Or we might have a situation where we have, maybe some resistant larvae, and these are the ones that might be able to, make it through those stressors and provide exchange. And what we want to understand is like, okay, if are these resistant genotypes, like where are they on the genome, and how is this affecting both the larval pools that are coming in the adult populations for them. So we do this in a two-phased approach. The first one is what I call the torture phase. We take early life history stages and we expose them to various combinations of stressors, and we look for death. We look for mortality, and hopefully finding selective mortality across some of these stressors. And using that, we're trying to identify loci that are responding to the stress. So basically, we're looking for allele frequency changes before and after exposure. So with that, we can identify certain loci that are under or putatively under selection in early life history. And we'll also get just random loci from the inefficiency of the capture that happens. And so with that, we end up with a set of capture probes derived from our early life history experiments. Then we go out and we collect adult samples across different environmental gradients. And the idea is that we can sort of separate out these early life history loci we might infer to be selection, like using outlier tests in adults, and neutral loci. And theoretically, we would have some different relationships between those stressors and genetic distance that are happening. So theoretically, we should have some stronger correlations between either these early life history ones and adult ones relative to our neutral loci. So this is sort of the basis of several projects that are going on in the lab right now. It just sort of switches between life history stage and stressors. And so to do this, I gotta highlight the effort of my team. We built a system from scratch. Building an oyster hatchery was a lot harder than we anticipated. And so this was a lot of effort from a ton of people, but we now have a computer-controlled system that we can replicate and basically have these diurnal cyclings in a predictable way in either static or flow through. We've been able to do this. And now we've also been able to adapt the system to do larvae and juveniles. So it's been a ton of work setting this up and getting it going in the lab, and with a ton of people putting in the effort. But now, we've been able to do this. So this is nearly eyed larvae. So these are nearly ready to settle. And then we've been doing this on just settled juveniles and a little bit older ones, doing these different experiments. So these are the life history stages we've been focusing on. And just to highlight, again, it takes a really large village to keep this going between collecting brood stock, feeding larvae. Is a multiple daily event, cleaning all this stuff. It's been a ton of people that have done this, and it's been a ton of work. And I'm telling you all this stuff, and I'm prefacing this because it's a ton of work, because I'm not actually going to show you any data from these experiments quite yet, but I am going to show you basically how this framework works with a couple existing projects that sort of predate this a little bit. But this is all what's coming, and this framework applies to what we're doing. And it's been an exciting time in the lab, since now we can actually raise oyster larvae, and they do what we want them to do, which is great. So to sort of give you a taste of what we're finding, I'm going to start with a project that I started as a postdoc in Katie Lauderhouse's lab. And this is the one that sort of generated all of these thoughts in making this whole experimental approach. And this one focused on coastal acidification and sewage effluent. And I already explained about how coastal acidification, epoxy are linked, and nutrient input, but basically this applies to sewage effluent as well. So this is a paper from 2014 where the folks went out and did transects along estuaries, looking at partial pressures of CO2. And you'll notice this hotspot of CO2, and it's right at an outfall of the sewage treatment plant. And this is basically can be a byproduct of either not removing nutrients from the sewage effluent, which happens, or it can also happen from sewage treatment, where we're using microbes to actually break down the nutrients, and they're respiring, then pumping out CO2. So it can happen in different ways and different levels of treatment. And in case you're wondering, sewage effluent is everywhere. Every one of those blue dots is a municipal treatment plant in the East Coast. Sorry, I don't have my West Coast maps up to speed. But the volume of this is staggering. It could be, several million or hundreds of millions of liters or billions of liters a day in different estuaries where effluent's coming in. So this is, a large volume stressor that's affecting nearly all coastal waters. Effluent can have all kinds of things in it. Farm waste from personal care products, can be osmotic stress, heavy metals, pathogens, all kinds of things. And I think it's pretty easy to imagine that any one of those could be something that's inducing larval mortality. So this was what we were kind of thinking. And we knew that it was tightly linked to the coastal acidification. So we decided to pair these stressors together and do a factorial exposure between the two. So we have coastal acidification, what I keep saying, is Ca, and then sewage effluent is Se, and then the multi-stressor of them together is CASE, which I'll say case. So if I say case, just know that I'm talking about the multi-stressor. So we wanted to know, is this, impacting mortality? And then can we see that, there's shifts in allele frequencies that are happening before and after exposure? So to do this, we basically spawned larvae, we spawned adult oysters and got a giant pool of larvae. We grew them up to the trochophore stage, so just about 18 hours post-fertilization, we would take multiple samples of this initial pool of larvae. Right after we take the sample, we transfer them to one of 4 different treatments. Our acidified water was about 2,800 microatmospheres of CO2, so really high, not really meant to be representative biologically. We're really looking for signal here. So pardon that, I know that number looks crazy high. Our sewage effluent treatment was a 5% mix of sewage effluent by volume in our containers. Then we had the 2 stressors together, and then a control treatment was just ambient seawater. We did this just for 24 hours, and afterwards we pooled the larvae outside of or from each one of the experimental containers, and then did EEC to look at the changes in allele frequencies. I'll start just by sharing some of the mortality data. This is oversimplifying it a little bit. Oh, and I should say we did this across, like main point, we did this across 3 different independent spawns to do this. So we have 3 different independent blocks of this experiment. Sorry about that. So I'll say that across just looking at mortality, the overwhelming pattern that we had is that the addition of sewage effluent caused larval mortality. There was a little bit of a difference between the multi-stressor and sewage effluent alone, but really we saw this pattern pretty consistently across the different replicates. The other thing that we saw was that really the acidification treatment wasn't really inducing a lot of mortality all on its own. There's a lot of different possibilities for this. Mainly it was a short duration trial, and so this could have just been something they could have handled. But it was still a little bit surprising to us in the end. But there was mortality, it just wasn't anywhere near to the same level that we were seeing the sewage effluent ones. So again, we used exome capture. We were able to get about 400,000 SNPs across our different blocks, and about 350,000 of them were in or with the near exon. So the exon capture was working pretty well in the way we kind of wanted it. So the first bit of genetic data I'll share with you is a PCA, and this is just from 10,000 random loci that we pulled without anything else. And I color-coded these by our block and spawn, and this is just to highlight that each one of these spawns is fairly unique, and they're highly related to each other. So me using PCA to show this to you is a little biased, because it really will pull out these differences. But I think it's a really important point to think about when folks are trying to consider doing experiments in these systems, because doing multiple spawns is really hard, and I think it's also really valuable because we have these large differences and you want to be able to see them across multiple spawns. So I like to just highlight that, how different each one of the spawns were. So to sort of detect these loci that are under selection, we did outlier detection using a modified CMH test. And this one is by Spitzer et al. From 2020. And it does a better job of accounting for variance, but really what we're trying to see is that we have our initial allele frequencies. We want to see change in one of our experimental treatments while simultaneously not seeing any change in our control treatments, because obviously putting larvae in small little experimental bottles is going to cause some sort of selection too. And so we want to make sure we're not capturing that in our data set. And so we did a fairly conservative FDR rate of about 5%. And we had to find signal in more than one spawn to consider this an outlier. So that was one of the key factors here. And so I'm going to plot some Manhattan plots here. So this is our multi-stressor treatment. And these are just the outlier loci. I'm not plotting the non-outlier ones. This was our acidification one, and this is our sewage effluent one. There's really only 2 things I want you to take home from these plots. One is that the p-values are a lot higher in our multi-stressor one, so we had stronger shifts in allele frequencies. The other thing is that we don't have any single strong effect loci here. Really a very sort of, what we might expect for polygenic or small effect loci that we're seeing basically crossing. This is the whole 10 chromosomes, the entire genome. So we're seeing these outliers in every single one of the chromosomes. So this is an interesting pattern, especially contrasting to what we've seen in some adult studies. Again, this is a PCA. This one's color-coded by all the treatments. Again, this is just the outlier loci, and still we have, a very strong relationship between where this, the spawn relative to treatment. But we can, break this down in between the spawns, and it actually starts to show a consistent pattern. So here is one of our spawning blocks, block 11. These light blue are our initial samples, and then these gray ones are our control. So there is some change that happens between the two, but we can see they're still the two closely related ones. This is our acidification treatment. This is the multi-stressor treatment, and this is the sewage effluent treatment. So we sort of have this stepwise difference, and then this differentiation between the multi-stressor and the sewage effluent treatment. So this is block 11. Block 12 has a very similar pattern. And then block 10, almost similar pattern. Don't get fooled by the weird polarization that happens with the PCA. But here we have much more of sort of a mixing between the multi-stressor and the single stressor of sewage effluent. And here we have more mixing between the controls and the coastal acidification treatment here. But I think for marine invertebrates, this is as consistent pattern as it gets. So I'm calling this a consistent pattern of change that we're seeing across our outliers. The other really great thing about using these shared outliers is that we really start to see, I think, a representative pattern of the change in outliers across our treatment. So this is if we stuck with my original characterization where we had to have a false discovery rate less than 5%, and we had to see it in more than one spawn. And here I think we see a pattern that makes some sense where our multi-stressor treatment has more SNPs that are all exclusive to that treatment, but it shares a ton of SNPs with the two single stressors. And we have a core of SNPs that are involved in all three, which I don't think is all that surprising. I say this because we first started analyzing just one spawn at a time. And if you do that, you get a totally different pattern. And you basically just get all of this driven by like the single exclusive stressor loci. And I think some of this is just the sort of the nature of the CMH test, and that it's probably a little bit too sensitive. And so I like having that ability to look for, this has to show up in more than one spawn, I think gives us a much more representative pattern, and one that makes a lot more sense biologically than just, oh, everything's different in everything. And I could go through the different single spawns and they all kind of follow this pattern. But I think this one makes a lot more sense. So just to sum up, we had about 3,500 SNPs that we identified as outliers from that 400,000 that we started with. They're in about 1,800 unique gene annotations. One thing, I'm not going to spend too much time on GO-term analysis because I think in oysters it's a little tricky to interpret. But one interesting thing that jumped out of this was that if we look at sort of enhanced GO-terms across these loci, this is from the acidification. Green is cellular component, and yellow is biological processes. So these are ones that are significantly enhanced across the genome. And I tallied each one of these numbers on the side to make it seem a little more apparent. So this was the acidification one. Sewage effluent had a number more than this, but then we actually had the most in the multi-stressor treatment. So still kind of breaking down and trying to decide how much I believe in some of these GO-term enrichment analyses, but I do think it's an interesting pattern that the multi-stressor shows more of this enrichment than the two single stressors on their own. Sewage effluent was really driving some larval mortality, and I hopefully convince you that really like having those additional spawns, the multiple spawns to look across, provides an important check statistically and also an important characterization of what we're seeing. Like these spawns are actually very different from each other. So yeah, back to the second take-home message. Coastal stressors are shaping the larval genomic diversity, and that multiple spawns are important. The last project that I'll talk to you about is one that's very much a work in progress right now, and it's been really exciting for us because of our experiment. And so this is the work primarily of a PhD student in my lab, Amy Zick. And so this is looking specifically at Narragansett Bay, which is the large bay estuary that's in Rhode Island. If you weren't aware, this is Rhode Island. It's neither a road nor an island, which I was just in Canada and someone asked me if it was an island, and I had to explain that it wasn't. But yeah, this is Rhode Island, and so we have been working exclusively in here with these oysters. And again, the cool thing about this is we took the probes from, and we collected adult oysters from these different locations across Narragansett Bay, and we hybridized them to those same capture probes to do our genetic analysis. With that, we did a ton of environmental data collection. We collected a variable based on sewage effluent volume and distance from an effluent source. We looked at temperature data, salinity, acidification, dissolved oxygen, and we tried to compile across as many different sources as we could, looking at fall and summer averages, minimums and maximums, all these things, and controlling this environmental data. That like in its own is like its own PhD. Metadata for environmental stuff is tricky. But one thing that's an issue with all of our environmental data in Narragansett Bay and a lot of estuaries is that it's really a north-south gradient across almost all of these. So there's a lot of collinearity between our variables. So to try to account for this, we did a PCA of all these variables and tried to identify sort of these groupings that here I have color-coded. So we tried to just break this down to one representative variable, means clustering across the PCs. So in the end, we came down to about 10 different variables that we tested. And then we also tested the first 3 PCs of those environmental data combined with the idea that we might just not be able to properly tease apart the collinearity between these two, or between these several variables. So with the adults, we ended up with less SNPs, but we had about 2,200 neutral SNPs. And then we used differentiation-based techniques, so things like PCA adapt, outflank, base scan, things that weren't looking at environmental. But we counted, we had 58 differentiation-based outliers. And again, we're looking to possibly see the stronger relationship between the environment and the outlier SNPs relative to the neutral SNPs. And to examine this, we used a tool called RDA, or redundancy analysis. And this is a really handy tool for exploring these multivariable states where you have, basically 2 different giant matrices of things. And so here we have a giant matrix of genetic or allele frequencies, and we have giant matrices of environmental data. And so RDA allows you to break this down, go through a linear regression, and what are these environmental variables that are possibly driving these patterns. It's a fancy way of doing correlations, but it's one that's better. It takes into account the non-independence between loci and variables. So the first thing I'll share with you is that the coastal stressors are not shaping the neutral loci, which was following our expectations. So here we have a horrible R-squared and a surprisingly low p-value, but non-significant one. And the only environmental driver that came out from forward selection was minimum salinity, but this really is not explaining any variance in our data set. However, if we look at those differentiation-based outliers, we had the 58 loci, and here we found that 2 of our 3 PC variables, so the composite of the environments, were driving or have a fairly high correlation with explaining about 40% of the variance from the environment. So here PC2 is largely composite of pH variables, though it's not exclusively pH, and PC3 is temperature. So these are the 2 that are pulling out as sort of driving this pattern. And then one other thing that we explored was looking at observed heterozygosity. So this is and here we saw another really interesting relationship between observed heterozygosity and that same PC2 environmental variable. So not only is we finding it in the RDA, but just, we're having basically higher heterozygosity in our less stressful sites. And there's a lot of air quotes and all of those things, but this is a lot of proxy that we're doing with this. But I think this is a really interesting pattern that we pulled out here. And then the last thing I'll share with you, which is literally straight out of R and into today's talk, where we're actually finally doing what I said. To our analysis. So here we took the 3,000 outliers from the larvae, 363 of them overlapped with the SNPs that we got from the adults, and we tested them in the same RDA framework. So for the 363, we have a weak relationship. I'm not gonna lie, that's not the R squared that I wanted, but it is statistically significant, at a fairly good level. So not the best application of RDA. You don't really want one variable like this. So we also explored this using a sort of look at these same variables when you know that it's just one variable driving the system. So we used the RDA. We only found the one variable. So now we looked at correspondence analysis. You'll notice this graph is very similar. It's just inverted. The correspondence analysis is just another way of breaking down dimensionality in the genetic data. And here you can just do a straight up correlation between that average salinity variable and the first axis of our correspondence analysis. And we come up with a much stronger R squared, but almost exactly the same p-value, which isn't surprising because the way we test this is almost exactly the same. Cool result that we're seeing, this strong correlation between average salinity and the patterns that we're seeing exclusively in the early life history stage. So those case larvae. You might be asking, well, wait, you didn't test average salinity in that exposure experiment. And I would say you're correct, we did not. But salinity has a large impact on the buffering capacity of seawater. And so this variable, while it is just one, it is definitely correlated to a lot of the other environmental things. I'm just super stoked that we got anything out of this. I think this is like needle in the haystack stuff. So I'm super excited. Again, this is as preliminary as it gets. We're really down by the different stressors. I think we might be able to get some cooler things from this, but I'm still super excited about it. So I wanted to share that all with you. So just to kind of recap, we did see a stronger association in our adult outliers than versus the neutral SNPs with the environment, which is great. Sort of fit our hypothesis. And as we could tell, we have those composite environmental variables are driving that structure in our outlier loci. And strong, we definitely have some relationship between our larval outlier SNPs and average salinity and possibly other environmental variables. And you'll see that the pattern is not necessarily the same across these. So hopefully I'm convincing you that connecting our life history stages is a obtainable goal and one that's important for understanding evolution in marine species. So with that, I'll be sure to plug Amy's work. She's got a great repository up with all of her stuff. She's graduating in May, so if anyone's looking for a postdoc, she's a good person to get in touch with. And yeah, I'll just come back to my take-home messages. Hopefully I've at least convinced you of 3 of the 4, if not all 4. And I'll thank. Then I'll take any questions. Yeah.
So this might just be my terrestrial bias and I have no idea how anything marine works, but can you talk to me more about these different broods or spawns? They seem really different. Is it from the same individuals in the same place? over the same season, or are they different spawning groups in different areas? Like, just how does this whole spawning thing work? Because it seemed like a big deal.
Yeah, so in our particular experiment, those were all different individuals for each spawn, or different brood stocks, so different spawners. So that sort of enhances that difference between the spawns. And again, PCA is not the best way to show that to you because PCA is always going to group together things that are related. So the fact that all those larvae are at least at the very least distant cousins, at the very worst siblings, that's going to really pull those things together. So it is a very biased way of showing that, but I do think it demonstrates that those spawns are genetically different. And so if you're just basing all of your inference on one spawn, then you might be missing part of the picture. And I think to me that's both a statistical thing, like being able to sort of check some of these p-values that we get from the CMH tests, which are surprisingly low sometimes, but also the fact that those spawns start at very different spots, and even though they start at different spots, we're seeing shifts in sort of like the similar pattern that's happening. So gives me a little more confidence in what we're seeing.
Thank you.
It's perhaps going to be an annoying question to answer. In terms of how EEC works, I'm used to looking at low coverage data that
Sure.
doesn't differentiate based on exons and based on your RNA. So when you do the RNA capture, you're doing that after an exposure to a stressor.
Yes.
And so the RNA is specifically associated with expression related to that stressor, but then you talk about using those same probes for looking at many other stressors. And I also I work in plants, which express totally different things in totally different tissues, as do animals. What tissues do you use? And then how do you think about the bias that might create in the data in terms of what is actually in the data?
Yeah. No, it's a great question. And it's both the best thing and the worst thing about EEC. It's great in the sense that, especially for what we do, where we're setting up specific experiments for stressors, it's helped that we know, okay, we're going to really try to boost our coverage for the things that are expressed during the stressor. And when you're doing it for 24 hours, it's a nice time frame. The other nice thing about EEC is that you can control the RNA that you put into that pool for your probe. So if you're concerned about, you're not serving as much of the transcriptome as you want, you can just, add different tissue types. You can, do different experimental treatments. And there's really no limit to the RNA that you put into the pool. So that you can kind of control for that a little bit, depending on what your exact goal of the experiment is. For us, it works really well because and I should say, for the EEC, what we did was we pooled all of the treatments together into one probe pool because we didn't want to necessarily miss anything. And the stuff that was happening in the control treatment is still important for us to know, okay, we want to make sure this is not in our outlier set because it's experimental design. So yeah, it's a double-edged sword kind of thing. Yeah.
Thanks for that. I have two questions. The first is, are you missing out on the importance of regulatory regions?
Yes.
And the second is, rather than sort of applying these thresholds for detection and then overlapping them, have you thought about just taking correlations between your treatments across all loci and seeing if that's stronger than what you'd expect? Can you get signal from loci?
Yeah. It's two things I think about a lot. So the regulatory region question is a great one. And I'll say initially the idea of doing the exome capture was the fact that we couldn't afford to do the whole genome sequencing for the pools at the coverage level that we wanted. And so that was the idea. It's okay, we can't do whole genome sequencing. We can't afford to do it. So what do we do? And so to me, the exome capture was sort of the next best thing that we could do in that situation. The other thing I'll say, and something that we're just if you're working with the mice version 27 of the exome panel, they still get only 60% efficiency. So that means 40% of the reads are still going to other parts of the genome. And so like really, something that, we're gonna start doing is sort of calling genotypes traditionally in our exon loci, since that we have tons of coverage, but then taking a low coverage whole genome analytical approach to the rest of the genome, because we get about like 1x coverage across the rest of the genome. So that all that data is still there, and I'm like terrible about throwing away anything, especially data. So like that's something that we're going to start looking at. So I don't think it's necessarily lost. It's not emphasized, but because it's just harder to do that low coverage analysis. But for what we're doing things in juveniles and adults, we can definitely do that that approach, and so we can kind of hopefully get at sort of a best of both worlds type situation. But yeah, someone asked me earlier today, it's like, oh, why did you exclude them? And I'm like, well, I didn't exclude the regulatory regions. It was just, this was one way to go about it. But yeah. And then in terms of the correlations, like, yeah, it's a really good idea. And it's something that I analyzing this data, struggled with level that we want to use, like, because you'd be surprised at some of the p-values that you come out of the CMH test. And that's why I really liked, I took a, I was like 5% is good, and then show it in more than one spawn. And that sort of like felt like a good cutoff, and it seemed like a reasonable number of SNPs that came out of it. I think there's like, different ways you could go about it. And I haven't, quite so, nor has this been peer reviewed yet. So I'd be curious to see, what other people think about, the choices that we made to do that. But I think, a lot of times, I should say the previous work that's done these type of experiments, it's just been one spawn, do the CMH, pick usually a lower FTR rate, because you get so many loci that pop up with the CMH test, and just go from there. So I like the ability, okay, we can check this with the multiple spawns. So yeah. Thanks. Yeah.
One more quick EEC question.
Yeah, more.
I'm wondering how well it perhaps reduces the bias for RNA-seq for overexpressed highly expressed genes. Are you still seeing a bias towards exons that have high expression levels, or is that reduced by your methods?
Like the normalization helps a lot, and actually one of in the project that I was showing that one of my PhD students is working on, we're actually testing like different levels of normalization to see, because the one thing about the normalization is it's the hardest thing to do in the lab, because and it's not that hard, but you have to like pipette something in a thermocycler, and it's just a little nerve-wracking to do it, because you're just like you have to do a heat up so that everything denatures, and then you're like racing against time, because once the double-stranded DNA repairs, the enzyme chews it up. And so that's how you sort of like take advantage of the overabundance and thermodynamics to reduce down the highly expressed transcripts. But we're not sure you actually need to do that, because the coverage, we didn't see a strong level in the coverage when we did the original experiment, or a strong relationship between the coverage of the two, because we still got coverage at some of those, even if you saw in the heat shock one, the non-heat shock gene, like even though there wasn't that much RNA coverage, we still had decent DNA coverage. So we're toying with that idea, because if we could get rid of the normalization and it doesn't matter, then that's just like one easier thing, one less thing to put in that super complicated slide to do. Yeah. But it tails off. So like when we plotted just like pure correlation between the probes and the coverage in the captured DNA, is like basically it starts linear and then just sort of tails off, where the coverage of the probes doesn't seem to matter at some point, that you just get a certain amount of DNA coverage. If that applies to everything all the time, I don't know, but at least in oysters.